Hello, friends. I'm very happy to participate in webinar on material science and engineering and technology to deliver a research fellow lecture, though we cannot meet each other face-to-face due to the COVID-19 crisis. I believe that the present the organizing committee members should have faced on a lot of difficulties with unexpected things in preparing this unexperienced online event. So before giving my talk, I'd like to thank all the organizing committee members to organize this online meeting very successfully under the present coronavirus pandemic. I'm sure that their dedicated efforts could realize this unusual but meaningful meeting. As far as I know, my talk is a little bit different from yours, so I'd like to share more time with you about my research goal and my research strategy, rather than my experimental findings that I have made in the last 20 years. So in the beginning of my talk, I want to tell you what the nanoconvergence would be with the two-dimensional solids like clay. And if you allow me, I will tell you my research goal in advance. Well, my final goal is to apply anionic clay as a drug delivery vehicle for developing new hybrid drugs. Therefore, the two-dimensional clay nanoparticles will be an important biomaterial with very high additive value in the field of nanomedicine compared to any other industrial applications. So I will explain you what the drug delivery system is, how we can realize the new drug delivery system based on the 2D clay nanomaterial as a delivery carrier. And finally, I'd like to share my synthetic strategy with you based on the clay intercalation chemistry. And the rest of my talk, I'm going to bring some experimental findings on cytotoxicity of anionic clay nanoparticles and interaction mechanism between nanoparticles and the cellular membrane proteins, and finally the targeting functions of our nanoparticles. And then I'd like to demonstrate how we apply the clay-based drug delivery system for chemotherapy, gene therapy as a proof of concept. And finally, I will summarize my talk with some conclusions. Well, according to the MIT Technology Review, appeared in 2006, as you probably know, there were two research fields of NT-BT convergence, such as nanomedicine and nanobiomechanics out of 10 emerging technologies, as you can see here in this slide. As I mentioned in the beginning, I'm going to tell you the importance of nanomedicine in the near future. And what is nanomedicine? Nanomedicine is nothing but a hybridized word between nanotechnology and medicine, which is now considered as an emerging new field. Nanomedicine comprises of 6 research fields, such as drug delivery, drug and therapy, in vivo imaging, in vitro diagnostics, active implants, and biomaterials. However, drug delivery is thought to be the most important field in terms of the number of publications and the patents applied in the world. Then why DDS? There are several challenging issues, such as, the controlled release, solubility and bioavailability enhancement, and taste masking for sour drug or bitter drugs, overcoming drug resistance, particularly for chemotherapy and gene therapy, and passive and active targeting, etc., and etc. And now let me start to tell you the story about 2D nanocarrier for drug delivery. Of course, there are many different sort of drug delivery vehicle. However, the most intensively studied drug delivery carrier would be the liposome. And what's the liposome? Liposome is nothing but, self-assembly of amplified molecules. Well, in the core, drug will be encapsulated in the core, and the external surface could be modified depending upon your research goal. For example, if you want to enhance the solubility, polyethylene glycol could be, conjugated on the surface of this core the particles, or if you want to enhance the targeting functions, antibody or targeting, molecules could be grafted on the surface, something like that, according to the drug delivery, strategy. And not only the liposome, but also the polymeric nanoparticles and polymeric micelles and dendrimers are, very frequently applied for the drug delivery carrier. And not only the organic or polymeric nanoparticles or drug delivery vehicles, inorganic particles such as silica or carbon nanotube iron oxide have been also applied as the drug delivery vehicle. But I am particularly interested in the two-dimensional nanoparticles such as the synthetic, anionic clay such as lead double hydroxide. Of course, there are many different sort of two-dimensional inorganic drug delivery carrier, such as natural 1-to-1 type clay or 2-to-1 type clay, or the lead double hydroxide as I just mentioned. And I think in order to apply this lead double hydroxide as the drug delivery carrier, it is very important to understand the crystal structure of lead double hydroxide. In order to understand the crystal structure of lead double hydroxide, it is really necessary to understand the brucite structure. In brucite, magnesium ions are octahedrally coordinated with the 6 hydroxide ligands to form an octahedron. And these octahedrons are bound one another by sharing the edges to form an infinite sheets. And these infinite sheets are stacked on the top of each other to form an lamellar structure. However, in the lead double hydroxide structure, the magnesium ions in the brucite, the magnesium ions are partly substituted by trivalent cation such as aluminum-3 or chromium-3, Fe3, etc. In such a way, the positive layer charge can be generated. In order to compensate the positive layer charge, interlayer anion can be stabilized in between the hydroxide layers. Such negatively charged drug molecules, therefore the negatively charged drug molecules can be, encapsulated into this lamellar structure by simple ion exchange reactions. According to the literature, lead double hydroxide has been well known that they are very biocompatible, and they have a very high anion exchange capacity, so that the anionic, drug molecules can be encapsulated into this lamellar structure, as I mentioned before. And at the same time, lead double hydroxide is insoluble in neutral and basic environment. However, lead double hydroxide are degradable in an acidic solutions. Yeah, that's why LDH can be rather applicable, well applicable for the drug delivery system. And then how can we realize the drug immobilized lead, lead double hydroxide? Let's imagine that you have a two-dimensional inorganic materials, as you can see in the left-hand side. And if you can somehow immobilize the inorganic or organic or biomolecules into this laminar structure to form an AB-AB or AC-AC or AD-AD hybrid materials. In such a way, I was able to prepare new inorganic-inorganic nanohybrids or organic-inorganic or bioinorganic nanohybrids. Based on the how can we realize the hybrid materials with such a heterostructure? Of course, there are many different ways of preparing two-dimensional hybrid materials, such as intercalative ion exchange reaction, intercalative propping open reaction, intercalative, co-precipitation reaction, and exfoliation and reassembling reaction, or calcination and reconstruction reaction, and pillar reaction. But today, I want to tell you mostly an advantage of intercalation reaction to induce one-to-one type heterostructure in hybrid materials. In order to apply the hybrid materials for drug delivery system, it is really required to understand the nanotoxicity of such materials. So we have, checked the nanotoxicity of a double hydroxide nanovehicle based on the MTT-assay and trypan assay and membrane damage test and hemolysis test. Well, as you can see here, in the first slide, we actually checked the cell proliferation based on MTT-assay, as I mentioned before, in 3 different cell culture lines, namely L132, Hep2, MCM7, those which are tumor normal lung cells, and human hepatoblastoma, cancer cells, and the breast cancer cells. And as you can see clearly that our inorganic particles are not toxic up to such a high concentration, probably up to 500 microgram per milliliter. And then we also checked the lactate dehydrogenase leakage test. What is the lactate dehydrogenase? Lactate dehydrogenase is a kind of a cytosolic enzyme. When the membrane is, damaged or broken, the cytosolic enzyme could be released out. So we can actually measure the amount of released the enzyme. As you can see here, clearly that the lactate, lactate dehydrogenase is not that significant, as you can see here, at up to 100 microgram per ml. And also we checked the hemolysis effect of our inorganic particles, and we found that the hemolysis is fairly negligible up to 100 microgram per ml. That's why our inorganic particle, layered double hydroxide nanovehicle, could be applicable for drug delivery systems. So we also compared our inorganic particles with other inorganic, inorganic particles such as iron oxide and SiO2 and a single carbon nanotube. And we found that out of 4 different inorganic particles, we found that lead double hydroxide is least toxic compared to other, inorganic nanoparticles. In order to apply our inorganic particle for drug delivery system, it is required to understand the intercellular uptake behavior of our inorganic particles. That's why our lead double hydroxide was actually immobilized with the dye molecules, such as FITC. And then we have actually checked the intercellular behavior of our inorganic particle, as you can see here in this slide. And after 2 hours, one can see clearly the very strong fluorescence inside the cell. And as the incubation time goes on, the intercellular, the fluorescence is very significant. However, if the FITC only treated up to 5 micromole after 8 hours, one can see a very slight fluorescence inside the cell, but it's not that clear, which means that our LDH nanoparticles plays a role as a drug delivery vehicle, as you can see here. Then actually, we have discussed with the molecular biologists to understand the intercellular uptake mechanism of our inorganic particles. And what is endocytosis? Endocytosis is a kind of process whereby large alien particles are wrapped with the plasma membrane and transferred inside the cell in the form of a vesicle and a vacuole. So according to the literature, there are several endocytic pathways in the mammalian cells. Some of them are pretty well understood and some of them are not well understood. But according to our preliminary study, the macropinocytosis is excluded. So we need to check clathrin and the caveolin-mediated endocytosis to identify the particle internalization route. To determine the specific endocytic pathways of our nanoparticles, the immunofluorescence microscopy and confocal laser scanning microscopy are often used. When the nanoparticles were treated in a cell culture line, the membrane proteins are assembled there to wrap the alien particles, as I mentioned just before, and bring them inside the cell through the membrane. But we wanted to know what sort of protein molecules, membrane proteins, are responsible for the formation of vesicle, clathrin protein or caveolin protein. Because the corresponding antibodies and anti-antibodies with dye, conjugated with dye, were available commercially, the cellular membrane could be assayed by incubating the cells with antibody, and at the same time, cellular membrane could be revealed by dye conjugated with a secondary antibody under permeabilized condition. The immunofluorescence microscopy image, as you can see here, that both the clathrin staining red fluorescence and the LDH green one overlapped each other perfectly, though the red immunofluorescence and caveolin was observed but faintly. No clear merged image could be seen, as you can see here in the right-hand side, when treated with the LDH fluorescence as shown in this slide. These results clearly suggests that the clathrin-mediated endocytosis is the principal mechanism for the cellular internalization of LDH nanoparticles. Well, caveolin-mediated endocytosis does not appear to be responsible for LDH uptake, as you can see here in this figure. And we also confirmed the clathrin-mediated cellular uptake of LDH particles by determining the involvement of certain accessory proteins related to clathrin-mediated endocytosis. Thus, we investigated whether the FITC LDH is co-localized with the typical endocytic proteins, such as dynamine and EPS15, and others. Dynamine actually regulates the endocytic vesicle formation in different endocytic pathways, and EPS15 is involved in regulating clathrin-coated feed assembly. And as a positive control, the internalization of FITC conjugated LDH was also compared to that of transferrin, because transferrin is very well known to undergo the clathrin-mediated endocytosis through the transferrin receptor. Well, we incubated the whole cell with the FITCLH, as I mentioned before, and stained them with the antibody against the clotting, dynamine, and EPS15, where the Alexa flow 594 conjugate to the secondary antibodies as the probe. And we found that the FITC-LDH was well co-localized with clotting, dynamine, EPS15, and EPS15 plays a critical role only in clotting-mediated endocytosis by associating with the plasma membrane receptor, the AP2 protein, which is essential for receptor-mediated endocytosis. As a positive control, we also compared internalization of FITC-LDH with that of transferrin, which is well known to undergo the clotting-mediated endocytosis through the transferrin receptor, as I mentioned just before. Well, intracellular FITC-LDH was highly co-localized with the transferrin label with the Alexa flow 594. This is well consistent with the results seen for clotting dynamine in EPS15, confirming the clotting-mediated internalization of LDH nanoparticles. So we were able to reconfirm that our inorganic particles could be internalized inside the cell through the clotting-mediated endocytosis. And after getting this experiment results, we discussed with the medical doctor, oncologist in the Catholic University Hospital, and we discussed with them to apply our inorganic vehicle for chemotherapy. So the oncologist recommended the anti-cancer drug, methotrexate, as the anti-cancer drug. Well, methotrexate is a folate, analog. Is normally used to treat a variety of cancers, including, osteogenic sarcoma, bone cancer, and leukemia, and breast cancer, and head and neck cancer by inhibiting the metabolism of a folate. But, however, MTX can be limited in chemotherapy by its very short plasma half-life and drug resistance and high dosage required for cancer cell suppression. In this study, a drug delivery system is proposed to overcome such a limitation. So when the drug molecules was once treated in the cell culture lines, MTX can be internalized inside the cell through the folate receptor site and reduced folate carrier site. And once MTX is internalized inside the cell, MTX can be conjugated to the dehydrofolate reductase, which is the enzyme involved in the folate cycle. When this the enzyme dehydrofolate reductase is is actually deactivated by the conjugation of MTX, then the folate cycle could be stopped. Consequently, the MTX inhibits the DNA synthesis and cell growth and finally drives the cell to death. Well, the effect of free MTX or the MTX-LDH nanohybrid on the inhibition of cancer cell proliferation was actually evaluated in the MTX-sensitive host cell and MTX, resistant host cells, respectively, as you can see here in this slide. Well, cell proliferation was measured with the MTTSA based on the fact that the yellow tetrazolium salt MTT is reduced by metabolically active cells, resulting in purple foams and crystals. When free MTX was treated, inhibition of cell proliferation remarkably decreased in MTX sensitive cells compared to, and MTX resistant cells compared to MTX sensitive cell compared to the resistant cell. Which means that MTX resistance results in decreased drug efficacy, as you can see here in this slide. However, I'm sorry. Whereas the MTX LDH, however, the MTX LDH inhibited both host cells and host MTX cells with the same degree. I mean, the sensitive cell and the resistant cell. It is therefore important to note that MTX plays a role as a dehydrofolate reductase inhibitor and dehydrofolate reductase level in host MTX cells. I mean, the resistant cell is abnormally overexpressed. This result indicates that the MTX LDH can play a role as an effective anti-cancer drug, even in the dehydrofolate reductase overexpressing host cells. Our previous reports demonstrated that the LDH nanocarrier are internalized into host cell via clathrin-mediated endocytosis. The fact that the MTX LDH selects the same entry to LDH nanocarrier, suggesting that the MTX intercalatively encapsulated in LDH lattice can completely modulate ligand receptor. It is therefore concluded that the clathrin-mediated endocytic pathway of MTX LDH is responsible for an enhanced cellular internalization of MTX, allowing the drug resistance to be bypassed. We present here the first example of a drug LDH nanohybrid system to overcome drug resistance. The MTX LDH hybrid system can be highly effective, in both wild-type host cells and the MTX resistance host cells in terms of inhibition of cancer cell proliferation, cellular uptake, and the intracellular retention. We also demonstrated that the uptake mechanism of MTX LDH in host MTX cells was mediated by the clathrin-mediated endocytic pathway, the same entry of the LDH nanocarrier only. It is therefore concluded that the clathrin-coated pit for the internalization of MTX LDH, which is completely different from that for free MTX, is actually responsible for accumulating high drug concentrations even in host MTX cells and bypassing the MTX resistance. Based on these in vitro experimental results, we decided to prepare chemically well-defined MTX LDH nanohybrid. It's a nanohybrid drug actually, with an excellent colloidal property to eventually be injectable for in vivo experiments. MTX LDH nanohybrid was prepared by co-precipitation method, as I mentioned in the beginning of my talk, and thus prepared precipitate was further hydrothermally treated to obtain a colloidal drug by controlling the hydrothermal condition, such as reaction temperature and time. Actually, reaction temperature was around 100 degrees C, and which was treated on hydrothermal condition for 6 hours. According to the X-ray diffraction analysis of thus prepared MTX LDH, the basal spacing of LDH was expanded from 7.7 angstrom to 21 angstrom upon encapsulation of MTX into the LDH layer. As you can see in the SEM image in the left-hand side, the average particle size of MTX LDH was determined to be approximately 100 nanometer, which is in good agreement with the particle size distribution results obtained from Zeta Sizer. The cross-sectional high-resolution TEM images of MTX LDH clearly shows a well-ordered layer structure. The interlayer distance between periodic lattice frames from the 00L planes was approximately 21 angstrom, which is an excellent agreement with the XRD results, as you can see here in the middle of this figures. To compare the therapeutic efficacy of MTX and intact MTX and MTX LDH, we performed a tumor growth inhibition test in a orthotopic breast cancer model. As you can see here in this figure, the tumor volume was actually controlled after intraperitoneal injection of PBS only, LDH only, MTX only, and the MTX LDH drug into tumor-bearing mice. The mean tumor volume of the PBS and LDH treated group on the day 32 were around 3,000 several hundred cubic millimeter, respectively. And following the administration of the MTX, tumor volume was reduced down to 2,000 several hundred cubic millimeter, representing a 27% reduction compared to the previous treated group. The MTX LDH had even stronger anti-tumor effect than MTX, with a mean tumor volume of 600 cubic millimeter, representing 31% and 74% reduction in tumor volume, in terms of tumor volume, compared with the previous treated group and the MTX treated group, respectively. And now here you can see the in vivo toxicity. So you can see clearly the survival rate of MTX LDH drug is determined to be 100% compared to the control. This result suggests that the MTX LDH significantly enhances the therapeutic effect and as a consequence increases the survival rate compared to the free MTX. This really consistent with the results of the biodistribution study, which will be discussed afterwards. Well, according to our biodistribution study, we found that our nanoparticles accumulate in the beginning, in the first period of time after the injection, our particles accumulate in the liver, but as time goes on, the particles were released out. However, we wanted to check the liver toxicity. For checking the liver toxicity, serum levels of AST, aspartate aminotransferase, and the ALT, alanine aminotransferase, were studied carefully. And as you can see in this slide, AST and ALT levels of LDH MTX injected mice were found to be in the normal range. In addition, histopathological analysis of hematoxylin eosin stained liver sections, as you can see in the right-hand side, does not show any morphological abnormalities related to liver injuries in any of the treatment groups. It is therefore concluded that the LDH MTX nanoparticle drugs does not induce any liver toxicity and inflammatory immune response after systemic delivery. Since the tumor to liver ratio is a very important indicator of both the therapeutic efficacy and the safety profile, the enhanced tumor-to-lymph ratio for MTX-LDH indicates its high potential as an effective and safe systemic chemotherapeutic agent if you compare with the tumor-to-volume ratio of MTX-only treatment. And we actually applied our this platform technology to the gene therapy. We actually immobilized antisense into our double hydroxide in order to apply for this gene therapy. In antisense technology, they synthetically produce the complementary molecules, I mean the small gene fragments, seek out and bind to messenger RNA, blocking the final step of protein production. mRNA is the nucleic acid molecule that carries the genetic information from the DNA to the other cellular machinery involved in the protein production. By binding to mRNA, the antisense drug interrupt and inhibit the production of specific disease-related proteins such as the viral infection and the cancer, cardiovascular disease, inflammatory disease, and genetic disorders. So we actually discussed with the biochemist who immobilized the antisense into our lead double hydroxide for gene therapy. The antisense MIG that our collaborator recommended was a small gene fragment, as you can see, the sequence of this single-strand DNA. It's a very short one. The length of this antisense MIG is around several hundred nanometers. And then we actually, after preparing the antisense MIG immobilized LDH, we checked the cell viability. So as you can see here in this figure, we have very successfully demonstrated the effect of cell viability upon incubation of antisense MIG LDH nanohybrid in cell culture line, which is actually HL60 cell culture line. The time-dependent inhibition of cell proliferation could be observed in the human leukemia cell treated with the antisense MIG LDH nanohybrid compared to the control. As you can see here clearly, approximately 65% suppression of cell growth could be expected, which means, again, that the layered double hydroxide nanoparticles really plays a role as a delivery carrier. Well, this paper was actually published in Angewandte Chemie a long time ago, and with our experiment results was pretty highly, they highlighted in the Nature News. And the bio-inorganic hybrids was also highlighted in CNN News in the beginning of 2000, and also our research results was partly, presented in the MRS meeting in Boston, and our research was also selected as the 8 innovative material researchers at that time. So after getting those results, a question was, however, raised how antisense MIG could escape from lysosomal degradation. It was therefore highly needed to understand intracellular trafficking pathway of gene delivery nanocarrier in vitro. According to the literature, various particle size of LDH nanoparticles have been studied to develop delivery systems without any consideration of particle size, dependent intercellular uptake mechanism and the intracellular trafficking pathway. We have, however, clearly demonstrated that the LDHs with the size of 50 nanometer to 200 nanometer could be efficiently entered into cells through clock-mediated energy-dependent endocytosis. Here you have the results of 50 nanometer sized LDH. Well, this 50 nanometer sized, the pristine LDH was synthesized by co-precipitation method, as I mentioned before. It is very simple. A solution of magnesium nitrate and aluminum nitrate was titrated with the NaOH and Na bicarbonate solution to maintain the pH around 9.5 with a vigorous stirring for 24 hours. For 100 nanometer sized pristine LDH, the titrated solution was then followed by the hydrothermal treatment at 100 degrees C for 16 hours. In order to understand more in detail for intracellular fate and trafficking pathway in the human cells, we have prepared two different sized 50 nanometer and 100 nanometer sized LDHs, and covalently conjugated with the green fluorescent dye, FITC. The external surface of the pristine LDH was modified by grafting the aminosilane, whose amine end was then conjugated with the FITC. Each pristine was heated at 100 degrees C for 12 hours under vacuum condition. After dehydration, anhydrous toluene containing the APS, aminopropyl triethoxysilane, was added to the dehydrated pristine LDH, and then stirred for 24 hours at 60 degrees C under nitrogen atmosphere. After washing and freeze drying, the APS grafted LDH was dispersed in deionized water, and FITC ethanol solution was then added. And then the mixture was, stirred vigorously at 37 degrees C for 12 hours. Thus prepared LDH FITC was collected by centrifugation, washed with water ethanol solution, and then freeze dried. Well, for immunofluorescence staining, the cells were incubated with the primary antibody and secondary antibody against the organospecific indicators such as EEA1, which is the early endosomal antigen 1, and F7, and the LAMP1, lysosomal associated membrane protein 1, and TND38, which is a transcoordinate network membrane protein, and Calexin, which is an integral protein of the ER, were actually used as a marker of early endosome, late endosome, and lysosome, Golgi complex, and ER respectively. After incubation for 1 hour at room temperature, the cells were washed with several times with the PBS and then incubated with secondary antibodies, conjugated to Texas red for 1 hour. And cells were then stained with the Taki dye for 10 minutes at room temperature in the dark. The cells were, then washed and visualized by using confocal microscopy. For quantification of co-localization between the nanoparticles and the individual antibodies, images were analyzed from different areas of the plate, and a percentage of co-localization was then calculated from the amount of the co-localized area between total green area and red area using curve option. In case of 50 nanometer particles were almost equally localized in the lysosomes, as you can see here in the right-hand side, equally localized in lysosome and the Golgi apparatus during 30 minutes to 6 hours, suggesting that they follow two different pathways with the same degree. One is typical endocytic pathway represented by the early endosome, late endosome, lysosome. And the other is the exocytic pathway presented by early endosome, Golgi, Golgi. In case of 100 nanometer LDH nanoparticles, LDH nanoparticles, 100 nanometer particles was primarily co-localized with the early endosome at 30 minutes, as you can see here in the right-hand side. And then apparently high amount was found in the Golgi complex compared to the lysosome after 24-hour incubation. This is an indication that the many of the particles are not go through through the well-known, exocytic pathways. Most of the particles seems to follow direct exocytosis pathway represented by early endosome, Golgi, Golgi, rather escaping typical endosomal lysosomal degradation, implying an important role of exocytic pathway in the trafficking of 100 nanometers. So, and now probably you were very suspicious how our antisense gene could be very effectively internalized inside the cell without any lysosomal degradation. But, here we have the answers. The 100 nanometer particles will really play a role as the delivery carrier for gene delivery, antisense delivery, because many of antisense will actually, escape from the degradation of lysosome. Well, as I already mentioned, the 15 nanometer particles select two different pathways with the same degree, such as a typical endocytic pathway represented by the early endosome, late endosome, lysosome, and the exocytic pathway represented by the early endosome, Golgi, Golgi. In case of 100 nanoparticles in the right-hand side, the direct exocytosis pathway represented by the early endosome, Golgi, Golgi is clear, rather escaping from typical endosomal lysosomal degradation. It is therefore concluded that the LDH particles, nanoparticles, with a size of 100 nanometer may have a great potential as a delivery nanovehicle for gene delivery due to their unique size dependent intracellular trafficking pathway with the endosomal and the lysosomal escapes. So we are now clear why the cancer cell was so significantly suppressed when we deliver the antisense MIG into our leukemia cell culture lines. Well, in gene LDH hybrid system, no in vivo applications using xenograft or orthotopic mice model were found in the literature, except our xenograft mice model study. Well, as demonstrated by us, the tumor is specific siRNA LDH hybrid system with both active targeting and passive targeting functions was studied in vivo. In this report, LDH nanoparticles, 100 nanometer in size, with a passive targeting function, were also conjugated with a folic acid and active targeting moiety onto the surface LDH to enhance the therapeutic efficacy. As shown in this slide, the xenograft mice model bearing KB tumor was intraperitoneally injected with the LDH FA siRNA, siRNA, and showed strong inhibition of tumor growth of about 66% compared to the mice group administered only with the siRNA. Such an efficient anti-tumor effect of LDH-FA siRNA in vivo could be explained by its enhanced selectivity of siRNA to tumor tissues compared to other organs due to the targeting functions of LDH nanocarrier, which was also highlighted in Angewandte Chemie a few years ago. Well, I'd like to summarize my talk. Well, we were very successful in demonstrating that the inorganic clay nanoparticles are mainly internalized into cells via protein-mediated endocytosis, and size-dependent intracellular trafficking pathway would be useful to design the target-specific gene delivery systems. And we are also very proud of preparing the colloidal drug, MTX-LH drug, as an injectable nanomedicine. The homogeneous particle size, biocompatibility, and high therapeutic value of MTX-LH nanohybrid system make it a promising injectable nanomedicine for chemotherapy. Thank you very much for your attention.